keeping the Austrian spirit alive here. Bettina came through on an Austrian program and was a fantastic graduate student. Are we calling you doctor yet, Bettina?
No, not yet. I'm working on it, hopefully in 2 months. So
okay, well, best of luck to you. Thank you.
Thank you for the nice introduction, and thanks again for the honor to give the talk now, and to the honor to be my external research, and I really enjoyed the time with you. And so today I want to talk about sex data in publications, and all the data I'm presenting today were recorded or were generated and analyzed at the SIBYLS Beamlab there. And the paper I want to talk today is resolving domain positions of cellobiose dehydrogenase by small angle X-ray scattering. And first of all, why are we interested in this enzyme cellobiose dehydrogenase? It is an electron transferring enzyme, and it plays an important role in the biomass degradation, or more precise, in the lignocellulose degradation. It's also found in biosensors application or in the production of biofuel, biosensors. So there are lots of application, especially in the field of renewable energy. But now to have a closer look on this enzyme, it is a monomeric enzyme which is extracellularly produced in the secretomes of food-degrading fungi. And the main feature of this enzyme is its 2 domains. It consists out of a small cytochrome domain, which is connected via a flexible linker of about 15 to 20 amino acids to a catalytic dehydrogenase domain. And shown here in color, the cytochrome domain has a hexacoordinated bound heme B as cofactor, and in the dehydrogenase domain, you have a non-covalently bound FAD. Those 2 cofactors are required for this enzyme to fulfill its function as an electron shuttling, because the electrons are generated here at the DH domain, where FAD is reduced to FADH2. Then an interdomain electron transfer occurs, where the electron is shuttled from the FAD to the heme B, and from the heme B it can then be shuttled further to, for example, biosensors or to lytic polysaccharide monooxygenase, which is then responsible for the cleaving of glycosidic bonds. And it's known that this CDH-LPMO system together really enhances the biomass degradations. But please keep in mind that for this electron transfer, it's only the way that the electrons are generated here at the DH domain. Then you have this transfer, and then further here. So the electrons coming here and leaving on this domain. And so in my project, we were interested in the mobility. And in former years, they really tried to crystallize this full-length CDH, and it was quite tricky because of the mobility. But in 2015, they succeeded with crystallizing it. And the 2 approaches they crystallized were from 2 different cellobiose dehydrogenase. One was the cellobiose dehydrogenase from Myrococcum thermophilium, shown here in blue, and the other one from Neurospora crassa, shown here. And as it stands out already here, you see 2 different conformations. The one was crystallized in a closed state, where you can see that the 2 domains are close together, as well as the 2 cofactors, indicating that here this interdomain electron transfer is favored. Whereas in case of the open state, you see that the cytochrome domain moves further apart, and the heme B is now orientated towards to the surrounding, so that it can interact with biosensors or LPMO or all other reactants. And this already brings me to the aim of this paper and this talk, because we wanted to resolve this domain position, so the mobility of the cytochrome domain, which seems to be important for the electron transfer, as I need the closed and the open state conformations in solution by measuring SAXS. And as mentioned before, I had the honor to visit the SIBYLS beamline in Berkeley, which is a very nice view up there. And the picture you saw already in Dan talk, where we have the SEC system, which is coupled with the MILES and the SAXS. And here is a scheme how it looks like that. At the beginning, you have the SEC column, and then after it runs through to the SEC column, it is splitted. So one part goes to a MILES UV-Vis Quells detector to determine the molecular weight. And in our case, we could really confirm that I have monomerics and no aggregations or anything. So all enzyme I was measuring was monomer. And the other part goes to the SAC, to a flow cell, where you can see here a close up. So it comes from one side, one tube in, then it goes through this kind of window, and then it enters in the other side. So here, due to the flow cell, you have a continuous flow, which makes the measurements quite easy. And then, which was also mentioned by Greg and Dan before, there are different tools to analyze it. And I use the tool Scatter. And when you upload it, you end up with such chromatograms. And here you can see again nicely that I have only one peak. And also mentioned in Dan talks here, you actually select here, I selected the region for the background subtraction. So actually the program is then subtracting this region from the peak and is calculating the P-von-R functions and so on. And now talking about the pair distance distribution function, also Greg introduced it to you shortly. I want to do it once more with the two models I showed you before, with the closed and the open state. And as a reminder for this P-von-R function, it's calculating by you pick one atom, then you measure the distance to all other atoms. Then you pick the next atom, and again you measure the distance from all other atoms except for the one you already measured. And this you're repeating for the whole enzyme or protein. And then a P-von-R function looks like that. So in blue, you see the P-von-R function for the closed state, which is similar to a globular protein, as you can see, kind of a Gaussian distribution. In case of the open conformation, you see it's a little bit different. First of all, this peak is a little bit shifted towards to lower distances, because here in this peak only the distance is within the domains. But I think it's really noticeable is here this shoulder. This shoulder is due to these long distances you have now between these two domains. So with this theoretic calculation, you can see nicely that even if it's the same protein, so you have the same sequence, due to conformation, you see a difference in the SAXS curves. And now I want to show you my data. And in the first question we wanted to answer was, how does this cytochrome domain mobility is affected by the pH? And therefore, we measured a series of pH 3.5 till 7.5 with the native MTCDH. And here you can see nicely that at the lower pH, so 3.5, 4.5, I have more this Gaussian distribution, and above that, so from 5.5 onwards, you see that here a shoulder kind of appears, indicating that at higher pHs, it seems that an opening or a movement of the cytochrome domain happens. This was kind of expected, because here I show you the electrostatic surface potential measurements. And you can see at lower pH, so at acid 3.5, the surface charges are positively charged. Then at pH 4.5, which is the theoretical isoelectric point of this enzyme, we have it more or less neutral, and all above it's negatively charged. We think that this brings it that at the acid pH condition, so with 3.5, due to this positively charged, it kinds of have attractive interaction, and at the higher pH we have the repulsion interactions. But now in the next step, to get a better idea about which conformations are present, we performed SAXS-based multistate modeling by using BILBO-MD. And this method works that you use a starting model. In our case, we use the PDB, but for the PBD in the first step, we modeled all the glycans to it, and then a bunch of different conformations were generated, and therefore the domains were kept rigid and only the linker as well as the glycans were flexible. And with this, we generated a lot sets of different models with different conformations. And then the program is calculating the theoretical P von R or scattering data of this functions of these models. And then it's combining which model reconstruct your experimental scattering data the best. And there is the approach that you can say you take only one model to reconstruct, or you can have a two-state model or three-state model approach.
Is I the only one seeing a frozen screen? It sounds like.
No, yeah, it's because it's.
Oh,
thanks. And yeah, but in our case, we found out that this two-state modeling approach is the best, so indicating that we don't have one conformation, but maybe some differences. And then what we did next to visualize it, we did the plot. So actually we took the best 50 approaches for all conditions and plotted in a histogram shown here, the frequencies versus the radius of gyrations. And here it's also nicely seen that acid conditions, we have a lower G value indicating a more compact structure, and at higher pH it's a little bit higher. And here now the models for the lower pH 3.5 and 7.5, which are the two-state model for the best fittings. And you can see nicely that here it's more the compact state. Of course, it's not the one crystallized, but it's still more compact than here, which is really extended. And beside, I'm showing we were using then the tool Multifox, where you upload your scattering data and the model, and then it really shows you how well your model is fitting it, which is also here indicated by the error-weighted residual difference plots. And in both cases, our models fit quite well. And then to be sure that it's really the difference is due to the cytochrome domain mobility, we performed a reference experiment where we measured the dehydrogenase domain separately. And you can, for the low pH 3.5 and the high pH 7.5. And here you can see that the H domain per se is not influenced by the pH, indicating that here the difference in the curves seems to be due to this cytochrome domain mobility. So also with this, we could prove that with this CDH at the acids, we have a more compact structure, and we see a movement of the cytochrome towards to more open structure at higher pH. And as I told you before, I did it with the native MCCDH, which was glycosylated. And in the next step, to confirm and prove our data, we deglycosylated the enzyme and measured it again. And here you can see, we see a similar trend that at lower pH, I have kind of a Gaussian distribution, and at higher pH, this shoulder appears. So the trend is the same, but here you can see it more clear. And also here we performed the SAXS-based multi-state modeling with the histogram and the models indicating the same. That also shows the opening towards increasing pH. So with this, we really could show this opening process. But now the next question was, what is if we add calcium chloride to it? Because it is known that calcium is stabilizing the close conformation. If we go back to this electrostatic potential measurements, just as a reminder, at the high pH, we had the negatively charged surface. And now it was, the hypothesis was, if we add now calcium chloride, which is positively charged, maybe it compensates this repulsion attractions. And so we performed these measurements now at pH 6.5, and in addition of calcium chloride, and the results are shown here. Where you see, again at pH 6.5, we have this shoulder, which we also showed with saw before. But now by adding calcium chloride with 50 and 100 millimolar, we can see nicely that this shoulder disappears and we again have kind of a Gaussian distribution. We confirmed this again by SAXS-based multi-state modeling. And here you can see nicely that at the pH 6.5 we have here, and we really see a more compact structure. But although we somehow have a little bit of fraction with a more extended, but the overall fit, show really a more compact structure. And to make it more clearly, we performed here again this reference experiments with the DH domain. On the first side to confirm that the DH domain is not influenced by the calcium. And in the second part to see also here that this peak here, or the disappearance of this peak, really contributes to the mobility of the cytochrome domain. And also here we performed this with the deglycosylated variant, where you can see again nicely the shift from the shoulder here appearing with the pH 6.5 without adding calcium chloride, whereas it goes to a more Gaussian distribution by adding it. And the multi-state modeling reveals the same, that here we see a little bit of compact state, and here you can see that here the frequency is really high. So it really stabilizes the close conformation. And now maybe someone came up with the question, we could show that it stabilizes the close conformation, but if you have already a more compact state, can it have an additive effect to a compact state? And to answer this, we measured all at pH 3.5, where we know that it's already in a compact state. And the results are shown here, where you can see again in purple the curve of pH 3.5, and in light blue and green the measurements. And you see here barely any differences. These small differences could be due to the glycans, as the glycans also have a different behavior by adding calcium. And the modeling approach is shown here, where you still see that they kind of look similar, also with the best fittings. And now to confirm that maybe our hypothesis is correct and that these differences came due to the glycans, we deglycosylated again, and here you can see that the curve with pH 3.5 and the one with the calcium chloride kind of really overlay each other. So this indicates that the addition of calcium has no additive effect if you have already a compact structure. And this is also again confirmed by the modeling. And with this, I'm already at the summary. I hope that in this talk about presenting the SEC-SAXS data, I could show you what you can do, and that it's a cool tool to use to study the mobility of a domain. In my case, we could show the opening of the cytochrome domain towards increasing pH values, that we could show that calcium stabilizes the closed state, so you have an influence if you have an open state, but that there's no additive effect, and also the effect of glycosylations. And maybe this last part was very interesting for me when I was working there, because I never thought about the glycans and how glycans can influence the SAXS measurements. And therefore, I want to give you a take-home message on how these glycans can influence it. And I performed some modeling approaches on the two PDB structures, because here with the empty CDH, you can see here in black the normal empty CDH structure, which is crystallized, and for the crystallization, they had it to deglycosylated. So here you can see in black this nice Gaussian distribution. But as the deglycosylated before, actually the position of the glycans were marked, and in this variant we have 4 different N-glycans. So in the first approach, the Manos tree were modeled to all of these 4 positions, and you can see already here with the green curve that the peak is a little bit shifted towards to higher values. And then in the next step, we did a modeling approach where we had this mannose tree and actually had another tree so that we have a high glycosylated CDH, and you can see again a little bit of a shifting. Because here, in this case, you can see that here the glycans are really extended to the surrounding. So actually then you get here some longer distances. But more effect has it when you go now to this NC-CDH, where we have the open structure, because again, with the deglycosylated crystallized form, you can see here this peak appearing. And in this structure, we have 7 N-glycosylation sites. So by performing the glycosylated with this normal mannose tree, I came to this structure. And the PDB is here, this orange one, where you can already see that only due to adding the glycans, this prominent shoulder kind of disappears. And if I now add the hyperglycosylations, you can really see that it now less looks like a global, also like a Gaussian distribution, even though it's kind of have this open conformations. The reason for that are the glycans, because in this case, the glycans are located at the linker regions. And as you can see here now, the glycans are also between these 2 domains. So maybe shielding this effect, or now the large distances are not there anymore. And so with this, I want to give you as a take-home messages. Take in mind that your protein may be glycosylated, because in our case, all the models we used before, also for the calculating, were deglycosylated. And at the beginning, we had troubles with getting the perfect match for it. As we found out, we had to add the glycans and it to really reconstruct our scattering data. And another interesting tool for us was, it is not known the exact glycan pattern for our enzyme. We use this modeling approach to get an idea what could be possible. So actually we modeled them, and then we took our measured curve and compared them to get a view of this. And with this, I am at the end, and I want to thank my group in Vienna and the whole SIBYLS beamline for hosting me and giving me this great chance. And last but not least, thank you for your attention, and I'm happy to answer your questions.
Thank you, Bettina. Questions to a user for SEC-SAXS?
Once again, sorry.
Question, Bettina. I know that Dan did all his experiments on that complicated enzyme under anaerobic conditions. I was wondering if you had to do your experiments under anaerobic conditions as well.
No, we didn't do it in anaerobic. We did it with aerobic. So we was thinking about doing it, but then we decided it worked without, so we kept it like those. Do
you think that it would be affected by whether the system is reduced or oxidized, these mobility?
That's maybe a good question. We never thought about this, because we thought, okay, we can see it, so it should be fine. And I mean, if we make it now with these conditions, it's the conditions you mostly find outside, or if you use it in application, it's the system you more or less use with air. So this was why we decided if it's working. But of course, it would be interesting to see if you measure it anaerobic, if you have some differences.
Real quick, when
sorry, that was a really nice talk.
Thank you.
I was wondering if you had any difficulties preparing your models for Bilbo MD so that they had like the cofactors and glycosyl groups present.
Yeah, with this, Michael was busy, because we used first this Bilbo MD and it didn't work out, and then we used this Bilbo MD2. And then Michael had to do some more programming stuff, especially for the Heme B cofactor, but it's working now. So anybody else has problems with cofactors, they have now the tool to do it. And then the other big problem was the glycosylation, because we did it first just with the normal model. And then we didn't manage to really reconstruct the curve nicely. And so then we started, okay, add glycans to it. And then we found out by adding the glycans, the fits were getting better and better. But yeah, these were the bubbles we came in with modeling tools.
Josh had a question.
Yeah, just those models you had when you had the cytochrome domain on different positions and different percent.
Yeah.
So is that like the model is the average of those two with those percent? Is that right?
Yeah, actually, you mean like, for example, here, yeah, this is actually, this was, you get then out of this modeling approach, you get the results, and these are the best fittings ones. And actually, yeah, this is the percentage and the G from these two models we are taking to reconstruct the curve.
So what she's got here on top is 2 models per, so there's 4 models presented here in total. And the cytochrome domain is represented more than once, or it is visible more than once. The main domain is superimposed.
Yep. Thank you. Well, I guess also just bigger picture. So is this just fundamental research of like the most optimal operating conditions of this protein? Were you specifically like, looking for that optimal pH? Is there like biological implications to it? I was thinking because I study plant tissues that more calcium and lower pH is actually more reminiscent of growing tissues instead of woody tissues. So I thought that was kind of interesting that maybe this fungus doesn't only wants to digest the woody parts and not the herbaceous parts. But that's just me being
I mean,
fun.
honestly, at the moment, this enzyme, we are not so really interested in this wood degradation. I mean, yeah, there's an important part. We are more in this biosensor part, because what we are, or a company is working now on, is to try to get blood sensors for it. So to measure the blood sugar, for example, for diabetes, and convert it to a signal, and therefore to find the conditions which is needed to get this electron transfer. But of course, it's also important for this wood degrading to see which is under which condition it's working and which you don't need. Yeah.
Okay, cool. Thank you. Yeah, very nice talk.
Thank you.
I'd just like to point out that Bettina visited us for a couple weeks. We usually operate with you guys mailing in samples, but there is an opportunity to come visit us for multiple weeks. The high throughput of our collections, both SEC and high throughput, it doesn't make sense to come with your samples every time. But should you want to use other, the SPR capabilities, maybe other synchrotron techniques, maybe learn synchrotron stuff, we would fully invite you to come and visit for multiple weeks, and can help set that up.
Maybe to put an in, I found it was a great experience to come to you, and even I learned a lot also with dealing with the user samples, and so it's a great opportunity to get known this technique.
All right. I've had